stringent, more reliable conclusion, then you could change your cutoff to either smaller value or sometimes a little bit bigger value if you don't have any conclusion, right? So this is actually the typical statistics analysis. And Dong also introduced the germ cell tumor very briefly. So it's actually next dataset we are going to release, and you might be able to explore the data. So for your assignment 2, okay, we're going to announce it later on. So for the lecture of this week, so we are going to focus on gene expression analysis. So you all know, so for biological samples, so one key feature is, so we know the level of gene expression for many genes. So if you're working on human samples, we could have either 20,000 genes or sometimes 60,000 genes if you have redundant gene names or have non-coding genes. So therefore, it's very important for us to understand so how to perform the gene expression analysis using the sequencing data, and also perform some statistical analysis trying to understand the gene expression data. So the plan for this week, so we are going to focus on the sequencing data, especially for the NGS and next generation sequencing. And then using the NGS, so we could do the expression quantification to understand the expression level of different genes. And then based on quantification, we can do like a more advanced analysis. So depending on the time and depending on our schedule, so probably I'm going to go deeper to the gene ontology this week or next week, probably next week. So I feel we don't have sufficient time to finish gene ontology this week. But let's focus on gene expression this week. So, and in the tutorial this evening, so Ran will introduce how to process the data on computer or on cloud, okay, using Colab. And maybe I don't have time today to finish the whole introduction of the normalization or like a statistical analysis of gene expression. But this evening, you might be able to have access to the code for analysis of the expression data from Ran, from pre-processing to like a normalization and to Z-score or some typical, I would say, beautiful figures like a volcano plot or something like you might see it later on. But you might not know the details. So on Thursday, I'm going to show you some examples and continue like working on the expression analysis data, okay? And like introduce the details of understanding the code from Ran and also different skills of normalization. So also meanwhile, so I think in either today or Thursday lecture, we will have self-reflection exercise. So for the purpose of help you to remember important concepts, so in the data analysis. Also, probably you can refresh your memory of understanding like some statistical analysis. So we introduced last week as well. So firstly, let's introduce how to process the NGS data. Okay. So when we are talking about NGS, so usually we are talking about like sequencing of DNA or sometimes sequencing of RNA. Okay, RNA. The traditional measurements of like DNA materials or DNA samples usually do the quantification of the amount. For example, we're always talking about the volume. Okay, volume of materials. For example, volume of DNA will be 23 microliter, right? So more comprehensively, we can also talking about the concentration, okay, concentration of the nucleotide in the sample. So usually, the very tiny samples, you could check what's the concentration of DNA or the concentration of RNA in the sample. It could be, for example, 34.6 nanogram per microliter, right? This is a kind of concentration quantification measurement. Or you could also talking about the binary signal, right? So whether this sample is virus positive, for example, SARS-CoV-2, positive or negative, right? So this is the binary signal. So these three descriptions are all like a quantification of a particular sample, right? So, but this kind of quantification is a very traditional way. So nowadays, if you are talking about quantification of a nucleotide sample, so we usually refer to sequencing, NGS, next generation sequencing. For the sequencing, usually we got the data more comprehensively. For example, we got this kind of data. We have the sequence of the nucleotide. So if it is a very long sequence, long DNA from human beings, then we could have very long sequence like this. So this is actually a sample from either A, C, or G, or T, right? We have very long sequence of such kind of characters. Okay, so for any samples, even the DNA amount is little, so we could have a huge amount of data. Okay, huge amount of data. So from those data, we can actually learning a lot of things. So okay, learning a lot of things. So here I'm going to share with you a video for the typical process of NGS. Okay, so start video.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with Elan, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3 prime ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next-generation sequence.
Right. So this video actually showed the technical procedure of the sequencing. So our analysis actually starting from the sequencing part. So once we got the sequencing data, so then we can do a lot of different types of analysis. Right? So because this technology is so standardized and I would say the technology becoming very mature. So many different methods has been developed based on the sequencing technology. So for example, here I show a list of sequencing methods. For example, the DNA level, you can do the whole genome sequencing, or exome sequencing, or ChIP-seq, or GRU-seq. So for RNA level, you could do the RNA sequencing, GRU-seq, PRO-seq. GRU-seq actually is to try to sequence a very fresh RNA, so just synthesis by the DNA. By sulfur sequencing can also help you to sequence the DNA methylation. The ataxi can help you to sequence the open region of the genome. And for this lecture today, we are going to focus only one type of sequencing, which is the RNA sequencing. Okay? So for RNA sequencing, actually, also there are many different types. So nowadays, the traditional way of the RNA sequencing is a bulk RNA sequencing. Basically, you got 10 million cells, right? So you sequence the cell using one experiment. You got the typical signal, or average signal from the a sample, right? So you got the expression level of different genes, so from different cells. And the single cell sequencing actually go one step further. So we will isolate the whole samples into a single cell level. Then you can measure the expression level of different genes in different cells. Okay, single cell sequencing. So more recently and very popular nowadays is a spatial, a single cell level, or sometimes it's subcellular level sequencing. So sometimes you could have for example, Stereo-seq could sequence the expression level of different genes in their nanometer level. But nowadays, becoming very popular and also generating a huge amount of data for biological samples. Okay. But today we don't have time to talk about too much details about other type of sequencing. If you have the interest, the AI tools out there. So other sequencing technology actually have a similar idea. So I'm going to share with you the major data format and the idea how to process such kind of data. And after you know about RNA sequencing, you will easily learn other types of sequencing by yourself. Okay. So the typical NGS pipeline, as shown in the video, basically you need to prepare a sample first. You got a sample, you purify the materials, either DNA or RNA, then you put your materials into the NGS instrument, for example, Illumina or some other companies, PacBio, provide machines help you do the sequencing. After the sequencing process, you go to have a lot of data, millions of reads from their nucleotide. Okay. Then what we need to do is a data analysis. Okay, data analysis. One type of data analysis in NGS is named as gene expression analysis. Because we know human has 20,000 genes, but at different conditions, in different cell types, the expression of different genes could be different. And those differences could be relevant to many processes. For example, if you are a biologist, you care about the human development, right? You may want to see, so in the development process, so how different genes could regulate the whole process, right? If you are a cancer biologist, you might have the interest to know what's the difference between cancer samples and normal samples. So here, show you one example. In the cancer condition, you got some cells or you got some tissue in the tumor condition. This is a melanoma, right? So melanoma samples, and you got maybe millions of melanoma cells, and compared with normal cells from the skin. You can then do some comparison. This comparison process need the whole steps of NGS. For example, you do the isolation of the RNA first, then you do the complementary DNA from the RNA, then you do the sequence, right? Then you can do the sequencing. After the sequencing, you got many reads from either samples, from both samples, you got a lot of reads. Then you compare the reads between the two samples, right? The question first is how can we do the comparison? Okay. Before the comparison, we need to understand. So in the typical sequencing protocol, so there are two types of sequencing methods. One is named as single-end sequencing. Another one is named the pair-end sequencing. So I also briefly introduced this concept. So during the introduction section, right? What's the meaning of single-end sequencing? Basically, for each fragment, this fragment is a real DNA fragment, either cDNA from the RNA sequencing or the real DNA fragments. Usually, this fragments is 100 base pairs, or sometimes could be longer, sometimes could be shorter. So anyway, this might follow the normal distribution with the length of this fragment, right? So on average, it's 100 to 700 BP, right? When we are doing the sequencing, we know like the sequencing reads usually have the fixed length. If you are talking about the typical Illumina sequencer, so the length of this read is about 100 BP. So 100 BP is for one read. Therefore, we got one question. When we are doing the sequencing, sometimes we cannot sequence the full DNA fragments. We cannot sequence the full fragments. We can only sequence from one end of the DNA fragments, and then we measure 100 BP of this fragment. Probably this fragment has 500 BP. You only measure 100, right? Therefore, you only see part of the fragment. Okay? To increase the visibility of the fragments, you could also do the pair-end sequencing. We know like DNA sequence have two strands, right? you could have the complementary strand in the other end. Therefore, you can design to sequence 2 reads for 1 fragment. So this 1 fragment actually can read from left to right, and also from right to left. But when you are reading from right side to left, you need to read the complementary sequence. Okay, you need to read the complementary sequence. Therefore, for each DNA fragment, you could generate 2 reads, read 1 and read 2. And these 2 reads are named the pair-end sequencing. Pair-end sequencing. Okay. So you can say one is colored in red, another colored in blue. There are some dashed lines in between, indicating that the middle part of this fragment is not sequenced by the sequencer. Okay. Then you may have one question. If in the middle part cannot be sequenced, how can we understand the sequence of the middle part? Any answer? Any idea? So this kind of sequencing, we can only sequence partially, right? Partially. Based on this partial information, can we know the full information from the DNA fragments or not? Think about it.
The sequence answer below. The sequence answer below.
So this is our DNA sequence, right? DNA fragment. And parent, right? Parent. We have 2 strands. So this is 5 primer. Right? A, B, long sequence. Okay. T, T, A, B. Sequence. We can sequence either this way, and we can do the parent sequencing. But in the middle part, it cannot be recognized in this fragment, right? How can we know the middle part? The answer is for each sequencing process, for each original DNA, we have actually many copies. We not only have 1 copy of the DNA, we have many copies. And then when we will do the fragmentation of the DNA, the fragmentation process is random. The fragmentation process is random. Therefore, so in the for 1, 2, 3, for example, I have 3 copies of the same DNA, right? I have 3 copies of same DNA. In the first copy, I cut in here, I got 1, 2, 3. I have 1, 2, 3. I have 3 fragments, right? For the second copy, the cutting position is randomly determined. Therefore, I might cut from here, from here, right? I got 2.1, 2.2, 2.3, 2.4. I got 4 fragments from copy 2, right? Similar for copy 3. Because I have millions of such copies of the DNA, therefore, I can almost guarantee that the middle part, although it cannot be captured in this fragment, it might be captured by another copy, right? Another copy. Therefore, I can read from here. It cannot be read in copy 1, but it can be read in copy 2, right? Therefore, this design can make sure we will understand the whole DNA sequence after sequencing sufficient number of reads, right? If we sequence sufficient number of copies of the DNA, then we can cover the full length of the DNA, right? This can be guaranteed, right? But from this citing, we can clearly know that the pair-end sequencing actually can cover more fragments of 1 fragmentation of the DNA, right? So here there are 2 reads cover 1 fragment, right? So after sequencing, so we got a lot of reads, right? I told you it's millions of, or 100 millions, or sometimes billions of reads for one sample, right? For one sample. So how can we put the data on computer? What's the most efficient way to store our data? So actually, a lot of pioneers design actually a format named FASTQ files. Okay, FASTQ files. FASTQ files using 4 lines to represent one read. 4 lines for one read. Okay, and the first line is the sequence ID. For example, this one, very comprehensive name, very long name. This is actually the name of that read. For each read, there is one ID. Okay. The second row, second line, is the real sequence. Okay. You can see it's always A, C, G, T, right? So this is actually the sequence of read 1. The third line, so for now it's meaningless, so it's a label, so it's a adding anything here, plus or something. Okay. And the fourth line is actually the indication of the quality of that base pair. Okay. Because when the sequencer tell you this position should be an A, okay, the sequencer also generates a p-value to tell you what is the confidence level the sequencer thinks this is an A. Okay, this is a p-value. But to representing a p-value, you need to give a very tiny number, right? For example, 10 to the minus 10 or 10 to the minus 3, right? It's a tiny number. How to represent this number by a letter or by a code, right? So this I'm going to introduce like the sequencing quality encoding. Okay? Sequencing quality encoding. So the method they're using is firstly translate or transform the p-value into a integer. Yes?
How do we know the length of the read? Like, is it random or?
The length of the read.
Yeah.
So it's a good question. It's actually related to the sequencing platform. It's not random. So typically they have equal length. So all the reads, so they have equal length. So the most commonly used platform actually have 100 BP. So Illumina, for example, they have 100 or 150 sometimes. Okay, depends on your machine. Okay,
So
it's a fixed length.
both of the strands should be complementary to each other? They should be complementary, right? The strands.
The answer is no. So this strand and this strand, so they are one, right? These are two. We're talking about this parent. So they are complementary to the real fragment, right? But these two, they are not complementary to each other. Yeah, they could far away from each other. Okay? Because the middle part is not covered as sequencing process. Yes.
How do you know which read is a forward read and a backward?
Yeah, this part could be guaranteed. So they have a label before the sequencing. So they know which part is R1, which part is R2. Okay. Okay, any other question? All right, so basically this is our document. If you open a, let's say, sequencing data, so it looks like this. This is raw data. Okay? This file could be huge. This is very big data set. Because you will have hundreds million reads. And for each read, you are going to have 4 rows, right? Let's say assume you have 1,000 reads. Then in this document, you are going to have 1,000 times 4 rows if you open it by like a text reader. Okay? 4,000 rows. Okay? And then very important value in this document is actually the line 4. Line 4. You can see this line 4, if you read it like a random, like a I, C, Z, F, F, E, F. It looks like a random, but there is a rule to determine the character in that location. The rule is actually determined here because I told you the meaning of this lecture is the probability of this A is correct, is not correct, okay? P-value, right? P-value. The probability is not an A, right? If this one probability, for example, if you is 10 to the minus 3, means every 1,000 statement, only one is wrong, right? If I read 1,000 positions, only one is wrong, but this is 10 to the minus 3, okay? 10 to the minus 3. But if we put 10 to the minus 3 as a location, it's very difficult to let us know that whether this is describing the first character or second character, right? We have to use a simple way to determine the p-value at that location, okay? That way is trying to transform the p-value to an integer, okay, to an integer. For example, we want to translate the p into a q value. Here we use very simple formula, 10 log 10 p, okay? For example, if p equals 0.1, 0.1 means so we have 10% of a chance making a mistake, right? This is a very big p-value. It's not good. But so if we put it into this formula, then we found out that so we got q. This is correct. Yes, this is correct. Then we got q equals 10, right? Q equals 10. It's an integer. And if a probability is 10 to the minus 4, we got q equals 40. Right? Then we got q value. Then the question now becomes, can we put the q value direct into this location? The answer again is no, because if the q equals 40, then the 40 will take 2 positions, right? But this is a very long read. It's for sometimes 1 position, sometimes 2 positions. This will making the document very complicated, right? Very difficult to matching them. Therefore, so people trying to use encoding technology to encode these quality values. The quality values could be from 0 to 40, right? But meanwhile, we also know that the ASCII code have also over 40 values, right? Therefore, for each ASCII code represents different symbols. There's so many symbols, you can listing all the symbols and given the ASCII code of each symbol, then matching the code to the Q score. So here, Q score 0 means ASCII 33 means this symbol, okay? Then you ranking all the symbols by ASCII code, and you got the Q score matching between the symbol and the Q score, right? This is how we got this kind of line 4, okay? For line 4, for each character here, you can check their ASCII number, and then matching the number to a Q score, then matching the Q score to the p-value. Therefore, each character here representing the reliability of this position. Okay, in terms of this is a G or a T, right? This is a matching process. Actually, the different machine and different sequencing platform are using different matching between the ASCII score and the Q score. Yes?
Shouldn't the quality score be only binary number like 1, 0? Because the sequence is like it's only one sequence, right?
It's not binary because you are representing the p-values, right? The p-value is a continuous value. You have to like a category, the p-value into 40 categories. 40 is the best, have very, very good quality, right? So therefore, if you are trying to represent 40 in terms of the Q-score, you need to use 73, and correspondingly, you need to use I to represent 40.
But if you're considering only one base pair, so the base pair should be only one pair, right?
Yes.
So
If you are only considering one base pair, but that base pair have a corresponding p-value, right? So you need to represent the p-value using one symbol. That symbol is representing the Q-score of that base, right? Understand? Okay. So here I'm share with you the typical platform, including the Sanger, so Lexia, Illumina, different versions of Illumina. So they using different encoding process. Let me take Sanger as an example. Sanger is S on the top, right? Sanger. If you are working on Sanger, this is the ASCII score. ASCII score from 33 to 126. This is the ASCII ID, right? ASCII ID of each symbol. Okay? ID of each symbol. And if you are talking about the Sanger sequencing, so 33 indicating 0. And I or 73 indicating 40. Okay? Remember what's the meaning of 40, right? 40, the Q score, then you should translate back to the p-value. Okay? 10 to the minus 4. Okay? 10 to the minus 4 in terms of the probability of this, this location is wrong. Okay? This is to the 40. You can also check other systems, for example, Illumina. If you are working on Illumina, then you look at the blue part. Illumina here, right? And the mapping between the ASCII number to Illumina Q score is here. 0 indicating H indicating 0, right? So H indicating 40. Okay. Understood? And yes. You don't need to use all the methods, so it depends on which platform you are using for sequencing. That platform will automatically give you those symbols to represent the quality from the sequencing. Right. Yeah, it depends on the choice of your platform. Yeah. Right. You got it from the machine directly. This is something that why you need to understand it, because some software have default setting. When you are checking the parameters of your software, sometimes they assume you are using Illumina. Therefore, they are checking this kind of matching. However, sometimes you didn't use Illumina, you use Sanger, right? So then this matching could be mismatched. Therefore, you might wrongly interpret the data from Sanger, right? Therefore, you need to understand that this matching actually depends on which platform you are using. And when you are running that software, you need to check whether the software is using the right parameters for the right sequencer platform. Okay? Right, after understanding this encoding process, here I'll give you one example. So you have the ID, right? The first row is the ID of your reads. Second row is your real sequencing, right? Then for each position, you use one symbol to represent the quality of this position, right? Then when you are reading this, like a quality score, the AAA, you cannot interpret this score if you don't know the platform, okay? Before interpretation, you need to ask which platform you are talking about, right? So if you are talking about the Illumina, you need to check the matching in the Illumina, the like a coding book of the Illumina, right? To say, okay, A in Illumina represent Q equals 40, for example. Then you're using the 40 to check back the p-value. Then you understand, okay, what's the meaning of A in this position, okay? If there's a high quality sequencing, you should always say very high, very small p-value, very high Q value in the reads, right? In the reads. And this is helping you to distinguish high quality reads versus low quality reads, okay? After your sequencing. Okay. So next step is data analysis. So once we got this kind of reads, what kind of analysis we can do? Of course, we can try to find mutation, for example. Let's say if for normal human, we always say GGGC, but for this person, we say GGCC. This is actually a mutation in that position, right? Because we could find it. So, but for the RNA sequencing, we can use this data to quantify the expression level of different genes, right? But before quantification of the gene expression, we need to do one further step. It's a mapping step, read mapping. Because this kind of read, if we know the species of the sample, for example, again, if we are working on human beings, we know human reference. You need to know a concept, it's the reference DNA. So we know like Human Genome Project, right? Eventually told us humans have 3 billion base pair in the DNA sequence. So those 3 billion base pair is already known. There is a book. So document the rough human genome, right? Rough human genome. So basically we know the whole sequence of human genome, right? So now we are sequencing one particular sample. We want to know where exactly the read comes from, right? So let's say if this part is very long, human genome, right? So then we got one read, R1, the only 100 base pair. So the question for mapping is where exactly this read comes from. This, for example, it's from here. This is R1. Maybe here is R2. Just for example, okay? So then the mapping process is trying to identify the location of each read on the human genome. The human genome. As we know, human genome is large. Therefore, we know the computational cost of this sequence mapping process actually is quite high, right? Therefore, you need to design really efficient software to solve this problem. So fortunately, pioneers already finished the steps. So this is actually one of the widely famous methods named STAR, can help you to do a very fast universal RNA sequencing alignment. For any given RNA sequencing read, the software could quickly tell you the location. Okay, tell you the location. So let's say you have 100 million reads. This software could complete the whole alignment in 5 minutes. Okay, really efficient. Okay, really efficient. So You don't need to understand the technical details, but if you do want to, you could read the paper and check the code, okay? They have very efficient code to help you do the alignment. But here, from a data scientist, if you want to understand the data, you need to understand the logic behind it. So in the RNA sequencing, the alignment steps is more complicated than the DNA sequencing. For the DNA, we know it's actually simply one-dimension sequence, right? One dimension only, one-dimension sequence. You just easily find the location of each read, then you're done, right? But for the RNA sequencing, you have to be aware of the splicing process. Because what we are sequencing is a matured RNA after the splicing. Therefore, quite often, so this read might not include the intron, and not include the intron. Therefore, you need to be aware of the structure of the intron in the RNA sequence. And this happens very often. Your reads could come from either the exon region. For example, this is the exon region of one gene named HMOX1. This is the exon region. You can see, so these whole reads are mapped to this location by that software. You find the location, right? But for some other reads, for example, this read, you can clearly see one read is cutting into two parts. Okay, one part is mapped to the one location of the genome. Another part is mapping to another location of the genome. Okay, this is actually named exon-exon junction. It's a junction, okay, between two exons. Okay, so typically, because we almost know all the potential junctions in human genome, so there is a particular pattern. For example, in the one side of the exon, you always see GU. GU is a donor site of the splicing region. In another side, you always see A and PY and AG in another side. Therefore, based on this kind of pattern, we can easily find all the junctions in RNA sequencing. Okay, then we can count how many reads are mapped to each location, right? This is mapping process. And this process has been finished by the software. We don't need to know the details, but we do want to know at least the name of the software for the alignment. Okay? So after this alignment, you might then wonder, so we have hundreds of millions of reads. Also, we have huge genome. What kind of documentation could help us to remember this read 1 is in this location, and this read 2 in this location, and all of the millions of reads are in each location. So we put all those data into another document named BAM file, BAM, or you can also name it a SAM file. SAM and BAM, they are very similar. So SAM is much bigger than BAM because BAM is the binary format. The SAM, you can open it directly from your text reader. Okay, it's unzipped, it's zipped. Okay, BAM and SAM, they are same thing, same format. So this BAM SAM format actually is after alignment. You want to know where exactly each read were aligned in the genome, right? So there are two components. This document looks very comprehensive, but actually it's very easy. So there are only two components in this format. One is named a header. Header is trying to describe the sample name, the sample length, and also the alignment method are used. Okay? For example, here I show you one simple example of the header in a BAM file. In this BAM file, we can see, so this alignment was aligned against the chromosome 1 to chromosome Y, 1, 10, 11, so 2, 20, 21, and X and Y, okay? All the human chromosomes were aligned. This is a reference, okay? And here is a method used. So in the header, there's description of the software named the top hat. Top hat actually is another software. It's actually one of the early a software for alignment before star, named top hat. Okay, so using top hat, and this is a particular parameters used in the alignment process. Eventually, you got this band file because of this reference and because of this software top hat. Okay, after the header section, the next section is a real alignment section. Alignment section is a little bit more complicated, including multiple fields, okay, including the read name, read sequence, the read quality, the alignment information, and also the text, and also many other information are included in the alignment section. Okay, so let me show you the details about this 11 different fields, okay, in this alignment document. So for example, this is the alignment section of one example. Okay, here each row representing one read. Okay, different from FASTQ file. FASTQ file, 4 rows for one read, right? So in the FASTQ, but in the BAM file or in the SAM file, so 1 row represent 1 read. Okay, 1 row represent 1 read. So you see this one have a truncation. Actually, this is 1 row because the screen is not wide enough, so I don't have the screen to show the whole row. Actually, so there is a they didn't change the row from here. Okay, so in this example, so the first column representing the read ID. Remember the read ID in the FASTQ file. Second column is a flag indicating the mapping status or mapping results of this particular read. I'm going to talk about flag later on. It's very interesting data to represent the mapping process against the flag. So keep this one for now. So flag. And after flag, the third one is actually the location, chromosome 1, the position 16230. Okay? We know a human has 24 right, chromosomes. So we check the chromosome ID, chromosome location, and this alignment told us that this read were aligned to chromosome 1, 16230 position. Okay? And next is a mapping quality, MQ, indicating the mapping quality. This is also a number to represent how good the mapping has been performed, right? So how good the read mapped to the reference genome, okay? Sometimes the mapping could have errors, could have either sequencing error or sample mutation or SNP, single nucleotide polymorphism, okay? So either way, but we do use this MQ to represent how good the mapping has been performed, okay? Then this is actually the mapping details named cigar. Cigar actually is a encoded ID for representing the quality of the mapping process. Okay, so 81 means 81 base matched. Okay, then there are 296. N is actually the jumping, so that part is not matched. And 19 matched. So another 19 matched. Basically, this is representing a junction. Okay, a junction. You can see 81 plus 19 equals 100, right? So that read has 100 BP, and there is one junction in the middle of that read. This is the meaning of the cigar. And equal means equals chromosome 1. So this indicating the pair and read mapping location. Okay. if the parent read in the same chromosome, different location, but this is the distance with the other side. So you can say the following is the read, the real read. Okay, it's the real read and the read quality and some other information. So they are all stored into this BAM document. BAM document. These are the 11 different columns. Okay, and different columns. There is one column very important. It's a flag. In the column 2, right following the ID. What's the meaning of the flag? You could say numbers in this flag. Okay, and actually, these numbers they are bitwise flag. So let me help you to understand what's the meaning of a flag. What's the meaning of flag? It's actually a very smart design. So if you open a website named like a Bro Institute, GitHub, IO, Pcard, explain flags, this could help you to understand the flag online. Okay, this is the website. Let me try to open it here. I copy this one and paste it here. Okay, this is the website for understanding flag. So in the document I showed here, you could see a flag quite often 83, right? 83. If I type in 83 here and click explain. So this website will tell you what's the meaning of 83. So you can see some check here, right? So read pair, read map in proper pair, read reverse string, first in pair. Okay? So this is a summary of this flag. Okay? So for this summary, you can see read pair got 1 score. It's 1. Okay? Read map in proper pair got score 2. Okay? Read reverse string got a score is 10. Is this real 10? This is actually 16. Why is that? Because all the numbers showed here is actually is in the 16 like counting rule. Okay, basically for any number in the second position indicating you have at least 16. Okay, and then similarly for each row here, actually is representing 1 bit of the information. This is a bitwise information counting. Okay, so the first one is actually 1. Second one is 2 to the power 2. Okay, 2 to the power 3, 2 to the power 4, and all the way to the end. Okay, therefore you could have a very big number to represent the selection of different items, using this bitwise counting rule. Okay, so let's go the other way. If I have 1 read, so it's a parent read pair, for example here, I can click this one. Then I will say this is the first, my read is first in my pair, and this alignment is let's say the read failed platform, and the alignment is not good. Then if I click this 3 description, then immediately I can get a score. It's 577, and this score will be the flag for this particular read. Similarly, if you got a read from the BAM file with a particular score, for example, 163, this is a flag score, right? 163. You got 163. You want to know what's the meaning of 163? You can click explain. Then 163 indicate the read are paired. They're R1 and R2 are read paired. And the read mapped in a proper pair. And their mate reverse strand. Yeah, mate is actually the R2, right? R2 is a reverse strand. Second in pair. Okay? This is actually the overall description of one given read based on the alignment document. Okay? And this actually is one-to-one matching. If you have any reads, if you can describe them by these sentences, then you can generate your flag score. Right? For example, I have one read is not properly mapped, is not primary alignment, and also the read is the PCR or the duplicate, then I got a score 1280. Okay? So now you understand. So for the second column in the BAM file, you got a flag, a number. The number is representing a description. For example, 83 could be explained as read pair, read mapped in proper pairs, and read reverse strand, and first in pair. Okay, this description together describe the overall qualities of the mapping of this particular read. Okay. Any questions for BAM flag? Yeah, it's not easy to understand at the beginning, but if you familiar with like a bitwise representation of the score, you will understand why people use such a comprehensive way to represent this flag. Because this is actually the easiest way and save a lot of space on your computer. Yes?
What is the meaning of this zero in the parentheses?
Oh, this indicating this is by 16, like 16, like every 16 number you increase one. Okay, yeah. It's not by 10. Okay.
Oh, this by 16.
Right. This is by 16. Right. All right. So after the alignment, once you got the BAM file or SAM file, then you can use a software named IGV Viewer. So I didn't put the software here, but encourage everyone to explore the software named IGV Viewer. Right. This is the software. Yeah, you can explore it by yourself later on. This software will have the human genome and any other species genome as well. For example, if I'm looking at one particular location, I can say this is human genome, right? So this is chromosome 4, 88317 to 459 830. So you can see some genes are in this location, right? For DSCF, single protein. So this is actually human reference genome, right? human respiratory genome. After we perform RNA-seq, and then we do the alignment, then we can see the peak here. This peak actually is counting how many reads can be mapped to this particular location based on the software I just introduced. Okay, so then using this document, using this file and the different softwares, you can then calculate the read or the coverage at each location. You're counting the data, you can summarize the mapped read in each location. So here I give you one example. So for example, for a particular gene, if I have to know the expression level, I'm going to quantify how much the gene has been expressed in this sample. I can count the number of reads within the gene, right? So here, for example, I can calculate gene A maybe have 200 reads. Gene B might have 300 reads. Therefore, from this simple counting, we might say, okay, gene B might have higher expression level than A. Is this correct? So from simple understanding, this is okay, right? However, you might also want to think deeper. For example, some genes are longer, some genes are shorter, smaller, right? Therefore, when you are calculate whether gene B have higher expression level than A or not, you should also consider the length of a gene, right? Also, you need to also consider the sequencing depth of your sample as well, right? Therefore, people design a method, for example, using this following formula to quantify the expression level of a given gene, named either you can name it SPKM based on the fragment per kilo per matched million reads, okay? Or reads, total mapped reads per kilo per mapped million reads. Kilo means how many K base pair of your gene, okay? A gene could have 10K or 5K or 1K, okay? Then you calculate how many K your gene have. Then you need to know the length of gene A, length of gene B, and you also need to know the total number of reads in your sample. Then you calculate the SPKM and RTPKM to know the expression level of that gene, okay, to quantify the expression level of that gene. So that I already answered this question. So the difference between R and F, R means reads, right? F means fragment. So it's a little bit different. So fragments means, so we can use two reads to estimate the fragments. And then using the reads to directly calculate the expression level. It's slightly different, but they are very similar to each other. Okay. All right. So lastly, let's do a simple test to help you to refresh your memory to understand better about what we learned today. So I think the quiz is already been open, right? So you have 5 minutes so to answer how many questions. I think you can start now. Very simple question. And there are minutes for 5 questions. First one is what's the non-hypothesis for feature exact test from last week? Well, most of you have finished, let me quickly go through the questions. They are very simple, right? So just to refresh your memory. So non-hypothesis of Fisher exact test. So the non-hypothesis basically assume that the two category variables are independent, right? So therefore we have the Fisher exact test check whether we can reject the non-hypothesis. So first Q, file for the Sandgren method, Q equals 20. So actually I used this example earlier on. So 20 means 10 to the minus 2 for the p-value, okay? And which sum flag bit indicate the read is unmapped. So from here, you can easily see the unmapped is actually, what is the read unmapped here, right? Click this one, you see 4, right? So you got 4. And the map quality in some most commonly stored, like also correct mapping quality. So then the gene expression one, if you do the calculation, so you will see the answer should be A bigger than C bigger than B. Okay, it's very simple, right? That help you to remember so what we learned. And in the evening, so remember to come to the tutorial, and Ran will introduce so further analysis based on the counting we obtained from here. The next step actually, so we need to do the expression quantification in a scientific way. We want to understand so what's the indication of the expression level of each gene. And also Ran will introduce several reusable code you can use in your future. So to do the pre-processing of the recount data, and then to generate simple figures for those analysis. Okay, see you in the evening.